Thank you for joining us for Biology Notes. Today's topic is bacteria. We talk about bacteria a lot, and we know that we are surrounded by them and covered by them. They're in our intestines, they're on our food, they're all over us and our surroundings. And it's easy to get kind of grossed out by bacteria, but it's also important to note that we do have some vital relationships with these microorganisms as well. Today we'll be examining the structures, functions, and the good, the bad, and environmentally important aspects of bacteria. Bacteria can be divided into two kingdoms. We can classify bacteria as either kingdom Eubacteria or kingdom Archaebacteria. Eubacteria are true bacteria, the common strains that we typically think about. E. coli, Salmonella, and these can be found in a variety of conditions, including in the human body. Archaebacteria, however, are more extremophiles. They love to live in extreme environments. Some are thermophiles, meaning they can live in extremely hot environments. Some are acidophiles, meaning they can live in extremely acidic conditions. These are not pathogenic. These are not disease-causing. They don't live within us. But they are some of the oldest organisms that we know of on this planet. Our next topic is bacterial structure. Here's a typical bacterial cell. You can use this diagram to label the picture on your notes. Here we see a diagram of a typical bacterial cell. We can see that its structure is quite simple. Bacteria are prokaryotic, meaning that they do not have a nucleus or any other membrane-bound organelles. Bacteria do not have mitochondria, do not have chloroplasts or ER or Golgi. You can use this diagram to label the picture on your notes. It's important to notice that even though they don't have a nucleus, they do have free-floating DNA, genetic instructions, as well as ribosomes, the machinery to manufacture proteins. Here we can see that the bacterial cell is covered by a plasma membrane, the phospholipid bilayer, additionally a cell wall, and in a minute we'll see what the pilus and remind ourselves what the flagella does. In terms of size, bacteria are larger than viruses. So if this is a virus, here's our bacterium. But notice it is much smaller than a eukaryotic cell. Prokaryotes are typically much smaller and simpler. One of the structures that we saw on the diagram was the cell wall, and eubacteria have cell walls made of a material called peptidoglycan. And there are two different arrangements of bacterial cell walls. One is called gram-positive and one is called gram-negative. And these differ in their layout of the peptidoglycan and their lipopolysaccharides. Now, why does this actually matter to us? Well, the structure of their outermost covering actually determines which antibiotics might be effective in treating them. Some other structures that we saw on the outside of the bacterial cell diagram were pili and flagella. We've talked about flagella before. Flagella are going to be the longer, more tail-like extensions that function in motility. It's the movement of these structures that allow for this cell to propel itself. The pili, however, are much shorter extensions, and these are going to help bacteria stick to other bacteria and to surfaces. Not all bacteria get to have pili, but they function almost like cellular Velcro in adhering to other surfaces and cells. Now, nutritionally, bacteria can be pretty diverse. It says here they can be photoautotrophs, chemoautotrophs, or heterotrophs. And that means that some use the sun's energy to make their own food, some use other chemicals to make their own food, and some are consumers. Bacteria can be either aerobic or anaerobic, meaning they use oxygen or not. But all bacteria are unicellular, or single-celled organisms. Sometimes bacteria clump together, sometimes they even form strands or strings of cells. Additionally, some bacteria can form a really important protective structure. It's called an endospore. And basically, when conditions get really tough, when there are harsh living conditions, like the bacteria is running out of nutrients, or it's starting to dry up, or the temperature is really high, the bacteria can form an endospore around their chromosomes to protect their genetic material from those harsh conditions. Later, when conditions get better, they can reemerge and go back about their normal metabolic activities. We mentioned that bacteria can form clumps or strands. Well, bacteria can also be 3 different possible shapes. The first is bacillus, or rod-shaped. Notice these are almost like ovals, elongated. The second possible shape is coccus, or round shape. These are nice and spherical. And the third possible shape is spirillum, and these are spiral shaped. These look like little zigzags. With all the bacteria around us and in us, it's no surprise that bacteria are constantly reproducing. They have an incredibly short generation span, meaning they live for a short period of time and they reproduce very rapidly. In lab conditions, E. coli can actually divide every 20 minutes, which means they can produce a colony of 10 million to 100 million bacteria every 12 hours. Every day we lose about 20 billion bacteria through our fecal material, and E. coli can reproduce quickly enough to replace those 20 billion bacteria every day. How do they do this? Well, bacteria reproduce asexually through a process called binary fission. Binary fission is an even simpler version of mitosis, where the genetic material is copied, the cell elongates, each side of the cell gets a copy of the instructions, and the cell splits. Notice that because this is a bacterial cell, prokaryotes don't have to deal with the nuclear envelope dissolving, and they don't even need spindle fibers to help direct the chromosomes. So binary fission is much simpler than mitosis. Binary fission does result in identical cells, or clones. But we know that not all bacteria are the same. So how exactly do bacteria end up different? How do they shuffle their genetic information? The answer is through genetic recombination. This is where DNA from 2 individual cells can combine, and this results in a great deal of diversity in bacterial populations. There are 3 different ways that bacteria can recombine their DNA: conjugation, transformation, and transduction. Remember how we said some bacteria have structures called pili? We said a bacterium could have a pilus that helps it stick to surfaces and to other cells. So the first process of recombination is conjugation. And this is where a donor cell or a host cell is able to grow a pilus, a bridge or almost a straw, over to another cell. Now through that pilus, through that little straw connection, the original cell can copy DNA and pass new instructions over to the recipient. This is recombination because it introduces new DNA into a cell. The problem for us is that sometimes cells carry instructions about how to resist antibiotics. And if those instructions are passed through a pilus into a new cell, antibiotic resistance can travel really quickly through a bacterial population. Another way that bacterial cells can pick up new DNA is through transformation. In transformation, bacteria get new DNA from their environment, oftentimes from dead neighboring bacterial cells. Here we can see a neighboring cell Although it's no longer alive, its instructions can be released into the environment, and those can be picked up by existing bacterial cells. This is recombination because we do see the cell taking up new instructions. Back when we talked about Griffith and the mice experiments, we said that instructions could be taken up by cells, and that was a perfect example of transformation. Sometimes transformation does result in increased virulence or an increased ability for a bacteria to make an organism sick. It's also a way that humans can manipulate bacterial cells in the lab. We can coax them into taking up DNA from their environment. A third type of recombination is transduction. And transduction involves delivery of DNA from a host cell to another using a virus. So the bacteriophage is a virus that infects bacteria. And notice that normally it's going to come along, inject its instructions, and ask the host cell to package up new viruses. Under normal circumstances, the virus is trying to package up new viruses. It wants only viruses to be made so that a whole bunch of viruses can be released and attack new hosts. Well, occasionally, instead of having viral DNA packaged up inside the capsids, every once in a while, some of the bacterial or the host DNA will accidentally get packaged up. So notice that when this little bacteriophage with bacteria DNA in it gets released, as it comes up to its next victim, instead of injecting viral DNA, it's actually just carrying with it bacterial DNA from the previous cell that was infected. So normally this cell would have been attacked by a virus and been taken over, but now this cell is simply receiving new bacterial instructions. This is recombination because it is getting new bacterial instructions. It's a mistake on the part of the bacteriophage, but it works out in the bacterium's favor. He ended up getting a new variety of genes. Bacteria get a really bad rap. We know that a lot of them can make us sick, and we call these pathogenic bacteria. There are two main ways that bacteria can cause disease. Some metabolize their host. They break down tissue of the host using bacterial enzymes, like tuberculosis, anthrax, our cavities that we get in our teeth, and Lyme disease. All involve bacteria who break down our tissues. Some bacteria, on the other hand, secrete toxins that make us sick. Botulism is caused by a strain of bacteria called Clostridium botulinum, and that toxin affects our nervous system. Staph aureus, staph infections, can cause nausea, vomiting, and diarrhea. And that results from bacterial toxins being released as well. We can fight pathogenic bacteria, though, through disinfectants, which kill bacteria on surfaces, antiseptics, which kill bacteria on living tissue, and antibiotics. These can be topical, these can be injections, or these can be swallowed in pill form to try to keep bacteria from reproducing or from getting energy. Some antibiotics even disrupt bacterial cell wall structures, which result in death of the bacteria. The first antibiotic was discovered in 1928 by Alexander Fleming. He was working in the lab and realized that there was a fungus in a petri dish that was able to inhibit the growth of bacteria. So he isolated it, he named it penicillin, and now it's used to treat bacterial diseases like pneumonia. Antibiotics are chemicals that interfere with cellular processes. So why are they ineffective against viruses? Good, because viruses aren't real cells. Since they're not cells, since they're not even truly alive, you cannot use an antibiotic to treat a virus. The problem is, we know that bacteria are evolving and many are becoming antibiotic resistant. Mutations happen. When we fail to take all of our antibiotics, we actually encourage the growth and reproduction of resistant strains. and some are actually becoming resistant to many antibiotics.